40 to 50 domains, that is loops, are formed. RNA connectors helps to form loops. Each domain or loop further coiled and supercoiled and reduce size to 2 μm in diameter. This coiling helps by positively charged HU proteins and enzymes like DNA gyrase and DNA topoisomerase first to maintain supercoiled stage. Let's watch another video to understand the concept of supercoiling.
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase 1, twists are introduced in the DNA molecule that cause it to compact by coiling onto itself. This is much like the process of coiling that you can observe by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more
Packaging in eukaryotes. This is our next topic. In eukaryotes, nucleus is well developed with nuclear membrane, nucleolus, and thread-like material. Histone proteins are rich in lysine and arginine. Residues are basic in nature and are positively charged. These histones make a unit of 8 molecules known as histone octamer. Negatively charged helical DNA wrapped around positively charged histone octamer, forming a structure called nucleosome. Nucleosome core is made up of 2 molecules. Each molecule have 4 types of histone proteins: H2A, H2B, H3, and H4. Let's watch the given image, in which left side image shows nucleosome while the right side image shows beads on string which appear when the nucleosome is formed. H1 protein binds DNA thread where it enters and left the nucleosome. One nucleosome approximately contains 200 base pair long DNA wound around it. Nucleosomes are thread-like stain bodies and look like beads on string. Packaging of chromatin at higher levels need additional set of proteins called non-histone chromosomal proteins or NHC. Non-histone chromosomal proteins, that is NHC. Additional set of proteins help in packaging of chromatin. Number 1, heterochromatin. These are genetically inactive type of chromatin. Then they are stains strongly and appear dark. These are 2 to 3 times more rich in DNA than euchromatin, while they are present near centromere, telomere, and intercalated. Second type of chromatin is called euchromatin, which is genetically very active and fast replicating. It stains very light. Non-condensed part of chromonema is present in this chromatin. Let's watch the given video to understand the concept of packaging in eukaryotes.
Did you know that the length of a DNA or deoxyribonucleic acid double helix in a typical mammalian cell is about 2.2 meters? This length is obtained by multiplying the total number of base pairs present in the DNA double helix with the distance between two consecutive base pairs. Now, the total number of base pairs present in a DNA double helix in a typical mammalian cell is 6.6 multiplied by 10 raised to the power 9, and the distance between two consecutive base pairs is 0.34 nanometers, or 0.34 multiplied by 10 raised to the power minus 9 meters. By multiplying these two factors, we get the length of the DNA double helix as 2.2 meters. Have you wondered how such a long polymer is packaged within a typical nucleus with a dimension of about 10 raised to the power minus 6 meters? To understand this, let us learn about the packaging of a DNA double helix, which is different for both prokaryotes and eukaryotes. In prokaryotes such as E. coli, the nucleus is not very well defined. However, the negatively charged DNA is not scattered throughout the cell, but is arranged in large loops, is held together by a few positively charged proteins in a region known as the nucleoid. On the other hand, in eukaryotes, the manner in which a DNA double helix is packaged within the cell is more complex. Eukaryotes contain a single molecule of negatively charged DNA packaged around a spool of positively charged basic proteins known as histones. The charge in a protein is decided by the abundance of amino acid residues with charged side chains. Now in this case, the proteins are positively charged as the histones are rich in basic amino acid residues like lysines and arginines, which carry positive charges in their side chains. Furthermore, the histones are arranged to form a unit of 8 molecules known as the histone octamer. Now around the positively charged octamer, a negatively charged DNA molecule is wrapped to form a structure known as the nucleosome, which is held in place by the H1 histone. A typical nucleosome has around 200 base pairs of DNA helix, and the nucleosomes present in chromatin can be seen as a beads-on-string structure when observed under an electron microscope. It is the nucleosomes that make up the repeating unit in chromatin, the thread-like bodies present in nucleus. Now, the chromatin fibers are of two types, euchromatin and heterochromatin. In a typical nucleus, euchromatin fibers of 30 to 80 nanometers in diameter are loosely packed and stain light. On the other hand, heterochromatin fibers of about 300 nanometers in diameter are more densely packed and stain dark. Moreover, of the two types of chromatin fibers, euchromatin is transcriptionally active, while heterochromatin is inactive. These chromatin fibers coil further and condense to form short and thick bodies called chromosomes
during the metaphase stage of cell division. In this way, a DNA double helix is packaged into a chromosome, which is further packaged within the nucleus.